So here we're going to talk about components of the electron transport chain. Now there's the Chemi osmotic theory, which was introduced by Peter Mitchell in 1961, and this was trying to help to explain how ATP is going to be made. And so there's going to be differences in proton concentrations, and these differences are going to enable energy to be extracted from oxidative reactions. Now we're first going to talk about the different parts of the electron transport chain. And so we have to consider acceptors of electrons. And some of the acceptors of electrons that we've talked about are NAD and NADP, as well as our flavoproteins, our FAD and FMN. So NADP is going to be linked to dehydrogenases in the cytoplasm, and NAD is going to be linked to dehydrogenases in both the cytoplasm as well as the mitochondria. Now, FAD and FMN are also going to be able to pick up electrons, but these are never going to be regulatory because they're always going to be associated with an enzyme. NAD and NADP are free-floating, and so these are going to be able to be allosteric regulators of enzymes in many different pathways, such as your TCA cycle and glycolysis, etc. Importantly, neither NAD or NADP can cross the membrane, so the NAD that's in the matrix of the mitochondria will never be able to cross the mitochondria membrane and enter the cytosol, and vice versa, NAD in the cytosol cannot enter the mitochondria. Now for the electron transport chains, there's going to be electrons that are going to be donated to enzymes, and these enzymes are embedded in the inner membrane of the mitochondria. And these enzymes, as they get these donated electrons, are going to be able to pump against a concentration gradient hydrogen. And then the hydrogen will be able to create a potential, electrochemical potential across the membrane, which will eventually drive the hydrogen back into the matrix of the mitochondria through the ATP synthase, and the generation of ATP will ensue. Now, there are going to be important transfers and molecules within the electron transport chain that are going to be able to transfer these electrons in order to pump the hydrogens across the membrane. We're going to have a direct transfer of electrons, and this can occur with iron-containing enzymes. We're going to have some that are going to transfer as hydrogen atoms and transfer as hydride ions. Now here we have part of the electron transport chain. We have complex 1, 3, and 4, and then our ATP synthase. So complex 1, your electrons will enter through NADH, and this NADH again is going to come from your Krebs cycle, your TCA cycle. And so if your hydrogens enter through enzyme 1, complex 1, which is NADH dehydrogenase, it will pass them through to coenzyme Q, which we'll discuss in a moment, and it'll go straight to complex 3. Complex 2 is not shown here. We'll talk about that in a moment. But if you enter through complex 1, you go straight to coenzyme Q, then to complex 3. And then from complex 3, you go to cytochrome C, and then to complex 4. And this is where your oxygen becomes your final electron acceptor to generate water. Now, electron carrier molecules are going to be important in the electron transport chain, obviously. We are going to have NAD and those flavoproteins, FAD and FMN. We're also going to have another series of carriers. We're going to have ubiquinone, which is coenzyme Q, and then we're going to have cytochromes, and cytochromes are going to contain those iron-containing proteins. We're also going to have iron-sulfur proteins in the electron transport chain. So first up is ubiquinone. Ubiquinone is lipid soluble, which means it can move around in the bilayer of the inner membrane. It's going to accept one electron and become a semiquinone, If it accepts 2 electrons, it's going to become ubiquinol. And so this is going to be a very small molecule, and again, it's going to be able to move around, so it's going to be a shuttle that is going to carry electrons through the membrane in between the complexes 1 to 3 or 2 to 3. And ubiquinone can carry electrons, or it can carry protons in the forms of hydrogen. Our cytochromes are going to have iron centers. And so you can see these are different types of iron-containing proteins. We have our protoporphyrin, which contains iron. We have our heme C, and we have our heme A. So these are going to have iron as prosthetic groups. These are going to have an absorption of light, so they can give off a color. And the 3 classes reflect differently with respect to their absorption spectrum. Heme A is integral membrane protein as well as heme B, and heme C is going to be covalently attached to cysteine residues, and this is going to be on the outer surface of the inner membrane of the mitochondria. So heme C is going to be what is going to be associated with the electron transport chain. Now there are also iron-sulfur proteins, and so iron is going to be associated with these sulfur proteins of cysteine residues, of course, and so you can see the cysteine residues. We have a risk iron-sulfur protein in which you have an iron atom which is going to be coordinated to 2 histidine residues rather than 2 cysteine residues, and so that's a risk iron-sulfur protein. And all of these are going to participate in 1 electron transport. Now, there are going to be at least 8 of these proteins involved in the electron transport chain of the inner membrane. So this picture shows all the components of the electron transport chain. We have our enzyme 1, which will pick up an electron from NADH from the TCA cycle. That's NADH dehydrogenase. This is going to be transferred to our flavoprotein FMN, which will transfer to an iron-sulfur center in complex 1. This is going to pass these electrons to coenzyme Q, ubiquinone, and then this will go on to complex 3, and then eventually to cytochrome C, and then to complex 4. So we have the first transfer here. We have our second transfer. We have our third transfer. We have our fourth transfer to our iron-sulfur centers. We have another transfer to our cytochrome C, and then finally to complex 4. Now when you enter through complex 2, this is part of the TCA cycle, succinate dehydrogenase, and in this case the electrons are transferred to FAD, and then you get a transfer to an iron-sulfur center, and then again you get another transfer to coenzyme Q or ubiquinone, and then it follows the same route 4, 5, 6. Now there are more steps. They're in between each of these complexes. Some have multiple iron-sulfur centers, etc. But this is just the big movements. We also have, of course, you're going to have different complexes that are going to bring in electrons from glycolysis, for example. We have our glycerol 3-phosphate shuttle, and that's going to donate to a enzyme that has FAD, and so that will transfer the electrons to that complex, which will then transfer it to ubiquinone, and we'll go down the route that way. And so the most amount of energy you can get from the electron transport chain is if you can get your electrons in the electron transport chain via the NADH dehydrogenase, because everything else skips that 4 hydrogens. So if you come in through complex 2, or if you come in through that glycerol 3-phosphate shuttle that we've talked about, you're going to skip that first step and you're not going to get those first 4 hydrogens pumped into the inner membrane space.